Mhm. Welcome to Dr. B.R. Ambedkar Open University. So it's MS second year Zoology course 7, and we have done with our unit 1. Today we have to deal with our unit 2, that is evaluation of insecticide activity. Earlier we have discussed about it, like the second unit we have done half of it, but now coming to this, the objective we will be learning today is to evaluate insecticides, the parameters and procedures for conducting toxicological investigations, and what are the different types of species of animals that we will be studying to evaluate the insecticide toxicity. And also we will do the statistical presentation of results using probit values. So coming to the chemical properties of the pesticide, we know each pesticide is unique, and pesticide is molecule is different in chemical structure, size, shape, stability, solubility, and electronic charge. Coming to solubility again, here we will be like the solubility is based on fat soluble or water soluble. So fat soluble will stay for longer durations in the I'll just check my screen. Yeah, coming to the chemical properties of the pesticide. So each pesticide is unique, and pesticide molecules differ in their chemical structure, size, shape, stability, and electronic charge. And these differences determine how pesticides function in the human body, right? So for example, the chemical structure of some pesticides make them fat soluble, while those of others make them water soluble. And the shape and electronic charge of some pesticide molecules allow binding to critical site receptors in cells or on cell membrane. So these and other variables often lead to significant differences in biological responses to pesticides. So once the pesticide enters into the body, so what will happen here? So once the pesticide enters, there will be a series of things that will take place. One is the absorption, distribution, metabolism, storage, and excretion. Again, coming to the absorption, so the pesticide can enter into your body through while you inhale the oxygen air, or by ingestion or oral food that you eat, or the water that you drink can be a contaminated water and all, and also through direct contact by skin absorption. So absorption is like a pesticides enter the body through inhalation, skin, or ingestion. And when pesticides are sprayed or applied across, they can become airborne and can be inhaled into the lungs. This can occur during the application of pesticides in agricultural settings or in the home when spraying for pests as well. And ingestion to mouth here, ingestion to mouth. So pesticides can be ingested if they consume like contaminated food or water, and consuming improperly washed fruits or vegetables, or drinking contaminated water. This can also lead to the pesticide exposure. And additionally, ingesting food prepared with utensils or surfaces that have been exposed uh, to some kinds of these chemicals can also lead to further toxicity. Okay, and dermal application like a skin contact or eye contact is also is there in pesticide encounter with the humans. So in dermal absorption, like through skin, so pesticides can enter the body through skin if a person handles pesticide-treated plants or soil or surface without proper protective equipment, or if there's a direct contact with the pesticide residue, the chemicals can be absorbed through the skin as well. And also through eye contact. If there is some splash of these pesticides while spraying, if they enter into the eyes, then also through that also the pesticide enters. So the site of exposure of the pesticide influences the rate of absorption into the bloodstream and its as well as its distribution pattern. Okay. So here we'll be dealing with the here we'll see like here you can see the how the pesticide is entering the body through different modes of entry, like ingestion, inhalation, skin absorption, which further leads to mouth, then it will reach the GI tract, and if it is like a water soluble, then it can easily be removed out, whereas if it is in fat, if it is a fat soluble pesticide or a chemical, then it will stay for longer durations, which can also lead to the toxicity. So this we will see in detail, but in order for you to remember it well, so you can remember the mnemonic like ADME, ADME, that which involves the fate of the pesticide as absorption, distribution, metabolism, and excretion. Yeah, pesticide absorption, distribution. Coming to the absorption, yeah, we have seen how the pesticide will enter and how it gets absorbed in the body. And coming to the distribution and movement in the body. So after absorption, what will happen? Pesticides are distributed through the body via the bloodstream. So their movement depends on the solubility of the pesticides. If it's a lipophilic or typically stored in fatty tissues, while hydrophilic are more likely to be found in blood and other aqueous environments. Come to plasma proteins. So many pesticides bind to plasma proteins like albumin, which help them transport throughout the body. So there are high chances of the pesticides to get transferred to different parts of the body. And certain pesticides, they can also uh, cross the blood-brain barrier, which will affect the central nervous system. And mostly the fat-soluble ones, only the fat-soluble ones, they can cross the blood-brain barrier and affect the central nervous system. So we have earlier discussed about few pesticides which are neurotoxic. So they come under these fat-soluble ones like organophosphate and all. So the distribution determines the potential organs and tissue affected by the pesticide. And coming to the uptake by organ, tissue, and cells. So once in the bloodstream, what will happen to the pesticides are they delivered to different organs and tissues. And these organs are like very crucial organs will also be included like liver, kidney, and the nervous system. So specific uptake, it depends on the affinity, like how much, the higher the affinity of a certain pesticide, so the more its uptake will be. And coming to storage, then fatty tissues tend to store lipophilic proteins. Lipophilic in a sense, they are like a fat soluble pesticides. So they have the tendency to stay for longer durations in your body if they are fat soluble, which will not be easily excreted through sweat, urine, or feces. And key organs for pesticides uptake. So the major organs which will uptake the pesticides are liver primarily. So as it involves in the metabolism, where the biotransformation of the pesticides, are not in the pesticides, but also the drugs and chemicals will, the primarily it will happen in the liver. Coming to the kidneys filter blood and some pesticides may be excreted in urine. However, pesticides may accumulate in the kidneys as well, causing kidney toxicity. And adipose tissues. So lipophilic pesticides, that are like fat-soluble pesticides, may be stored here for extended periods of time. A nervous system, I mean, the pesticides which are affecting the nervous system, some pesticides, especially neurotoxic ones, accumulate in the nervous system as well. The metabolism within the cells. Once inside the body, pesticides undergo metabolism, typically in the liver, which can alter the mechanism structure and make them more water soluble or less toxic. So metabolism majorly occurs in two phases. In the phase one, the functionalization happens, and in phase two, the conjugation happens. So in the phase one, we will just see in detail what will happen. So it will majorly happen here in the liver. So there are like hepatic metabolizing enzymes. So enzymes mainly SIPs, that are cytochrome P450s. They introduce certain functional groups into these pesticides like hydroxyl and amino groups, which will make the pesticides more reactive or water soluble so that they can be. So just look at the picture here. Here, this metabolism enzymes, majorly SIPs or cytochrome P450s. Here the enzymes, they will add the hydroxyl amino groups so that the pesticides becomes more water soluble, and it will be excreted through sweat, urine, or the bile juices that are secreted by the liver. They also helps in the removal of them through fecus. So that is one of the phase one metabolism that happens in the liver. And coming to the phase two, another phase that happens in the metabolism of the liver is like the pesticides or its metabolites are conjugate in the sense they are like chemically linked or attached to larger molecules like glucuronic acid and or sulfate, which are like water soluble. So water soluble molecules. So what they do, they go and bind to the pesticide, and then thereby they enhances their water solubility capability and make them get solubilized and then make them easily get excreted from our body. So these are the like the very important metabolism that occurs, or the liver, which plays major role in eliminating the pesticides, that is like detoxification or biotransformation we call it. So this metabolism can lead to detoxification. In many cases, pesticides are rendered less toxic while they are excreted, mostly water-soluble ones, as we have discussed earlier. But in some cases, this process, the phase one and phase two process in the liver, they do not only cause the detoxification of the pesticides, but there are certain pesticides which tend to get activated. They don't get detoxified and get removed, but in some pesticides, like propesticides, they require metabolism so that they become more active and they cause more toxicity to the different organs. And toxic metabolites, in some cases, the metabolites may be more toxic than the parent pesticides. So rather than the main pesticides, so whatever the compounds they are getting at the metabolites that comes from the main pesticide, they will be more toxic than the original one. Yeah. And coming to storage sites within the body, so pesticides, especially those that are fat soluble, can stay for longer durations, and this storage can lead to chronic exposure or long-term effects as pesticides are released slowly over the period of time. And the major storage uh, sites are fat tissue, bones, cheek, and liver. For fat soluble ones, and fat tissue for fat soluble ones, obviously, and bones and teeth, certain pesticides like lead, cadmium can be stored in bone tissue as well. Whereas in liver, pesticides are metabolized in the liver and may remain there, leading to liver toxicity. If they are not excreted, then there are higher chances of them to be remained in the liver. So it causes hepatitis toxicity or liver toxicity. And coming to the excretion and elimination from the body works to eliminate the pesticides through various excretion pathways. Like we have the first defense is our body, right? So it has different pathways to process the pesticide, and it has also different ways to excrete the pesticides out of our body. So majorly they were like urine, fecus, exhalation, and sweat. So through urine also so through urine the kidneys filter out water soluble pesticides, metabolites, and excreting them through urine. And fecus, some pesticides are excreted through bile into the intestines and then excreted via fecus, right? And exhalation, so when you exhale in the sense when you send out the breathing out, right? If you breathe out, there are chances of volatile pesticides to get excreted. And through sweat, some trace amounts of pesticides may be excreted through sweat as well. So the rate of elimination depends on the pesticide solubility. I mean, the process or the speed of the elimination and how they eliminate, it majorly depends on the solubility of the pesticides. If it is a lipophilic pesticide, then it happens in a different way. If it's a hydrophilic pesticide, then it happens in a different way. And the efficiency of the body's detoxification and excretion process is also based on the rate of elimination. So we are done with this, where we have seen the absorption, distribution, metabolism, and elimination. And we have also seen the storage sites and excretion process. So now we'll see the concept of bell curve here. Yeah. Is a bell curve, right? Yeah. So the concept of the bell-shaped curve. So what is a bell-shaped curve? So we know that different individuals have different sensitivities. Different individuals, they will have different sensitivities, and different species will react differently to particular compounds. So all individuals do not respond in a same manner, because for example, you take penicillin, it is a life-saving antibiotic for most people. However, some hypersensitive individuals will be there, which will, for them, it will be like, there will be side effects will be like too bad. All living organisms exhibit a broad spectrum of reactions and sensitivities to chemicals, but some people may be able to tolerate high doses, while others may be very susceptible to even low doses. So most individuals are somewhere between very resistant and very susceptible, right? So so this is what happens here when the sensitivity of large animals are graphed against increasing doses of chemical, the resulting distribution curve will be like this. So we will get a bell-shaped curve. It is called also normal distribution or Gaussian distribution on the honor of the scientist Gaussian, who have given this curve. So when the sensitivity of large animals are graphed against increasing doses, then you get this graph like this. So it represents the small number, the far, so this left and the left and that represents a small number, whereas the middle one, the majority of the population will fall into this, whether they are like susceptible or not. And here it falls into the majorly who was like very sensitive to the drug. And here this tail, it falls into the individuals who are like very resistant to the drug, but to the pesticide drug. But majority of them, like 99.5% of them, will fall into the middle, that is the central of the curve, where it is mostly suitable for 99.7% of them. Thus, in any toxicological test, all animals are not affected, and also that are affected will also exhibit certain types of relations. Okay, a toxicity to the drug. So let us consider like this is a graph. So on the y-axis, you have percentage population responding, and on the x-axis, you will have the doses. So the lower dose, no, nothing will happen. There is no lift in this. And in the middle dose, if the dose is like in the middle of it, because the lower doses, they don't produce any effect. We don't see any effect here. In the middle, we see the maximum effect, like how people are reacting to the drug. And when we go to the higher doses, the individuals who are like resistant will be seen here, and here the susceptibility individuals will be seen, and majority of them will fall, and a large exposed population will fall in this. The large exposed population will fall in this. So this is the medium dose. Graphical representation. So this is the graphical representation, and basically the bell-shaped normal distribution curve is the curve with a symmetrical. In normal distribution, the mean, median, and mode are located at the same point or at the peak of the curve. And here the standard deviation determines the spread of the data. So here the 68% of the data point is within the one standard deviation. You can see the standard deviation, which is -1. This is the standard deviation from the mean. Whereas 95% of the data fall within the second deviation from the mean, and 99.7% of the data or the data points lies within the third standard deviation from the mean. So this is known as 68, 95, 99.7 rule, or the empirical rule. And the the curve extends infinitely in both the directions but never touch the horizontal axis. This indicates that while extreme values are very unlikely, they are still possible to occur. Okay. And coming to the concept of the dose response. So we have seen that, so depending on different doses, so different population will respond to different doses, and 99.7% falls under the large exposed population. So here the concept of the dose comes here, where the toxicity of a pesticide is determined by quantifying the response of lab animals to a series of increasing concentrations or increasing doses of the concentration. So here, so let us look into this curve here. This is the relationship between a concept of dose and response, the typical dose and how they respond. So here response is seen in the percentage and here the dose. So if you keep on increasing the pesticide dose, so if you give 5 mg per kg of body weight, then you give 10 mg. If you give, I mean, you will check where it is causing the toxicity. So where the how much of the concentration is like not lethal or how much is lethal. So which is the lowest value, which is the highest value, and which will cause LD50. So in order to know the pesticide, whether it is toxic or not, you have to do this dose response curve analysis, or you have to see this concept to understand for the better understanding of the pesticide and to recommend the doses of uses of this pesticide, right? So this relation between administered dose and animal response is graphically depicted as the dose response curve here. And for a measured response such as death, the percentage of animals that die increase proportionally as the dose increases. So here we will see the NOAEL. So this indicates that no observed adverse effect level. So this is the highest dose of a substance that does not cause any harm or any effect. So till here, so till 10 mg, if you see, you can see that there is no raise in the peak, which indicates that there is nothing like difference or no effects can be seen in the patient or the organism that has taken up this pesticide. But however, after this NOAEL, here you see the lowest observed adverse effect level, right? So, and as exposure increases, the pesticides start becoming toxic, which further, upon further increase, can damage the nervous system, liver, kidney, or endocrine system. And here, see clearly here at 50, and here in between 20 to 25, like literally falling between 23 to 24. So 23 to 24% mg of the concentration of the pesticide causing the lethal dose 50. So lethal dose 50 means the lethal dose which causes 50% of the population to die. Right? We have studied in our early class what is LD50. So it will fall here. So once the lowest observed adverse effect level has increased, then here you see the LD50 value, which is a dangerous dose level. So increasing the dose beyond the threshold level increases not only the proportion of animals that they show response, but the severity of the effect as well. So it is this linear region of the dose response curve, which indicates that increase the dose, then increase the response. That is used to measure, describe, and predict the toxicological properties of a pesticide. And the third part of the curve here we see is the plateau curve. Plateau means the point where an increase in the dose is no longer produce an increase in response. So here, however, if you give drug, the plateau stays here, the highest point on the curve. The plateau begins at the point where an increase in the dose will not cause any effect because there the it has come to the end, the saturation and the receptors have been full, or the death of the animal might have occurred. So which will not take up further dose or further drug into it. So there is no response, even though if you increase the drug. So that is the plateau. So plateau will occur this. And here there is no raising up, but it will slowly tend to remain the same. And there is a something like Emax, like maximal effect, where the highest point on the curve indicating the maximum effect the substance can produce, even if the dose is increased further. And saturation of the receptors. So in many cases, plateau occurs when all the available receptors or biological targets have been fully occupied or saturated by the substrate. And the toxicity threshold is the plateau phase might also reflect the point where the system has reached a threshold. Means that is the maximum toxicity it can give, or if you go beyond that, then there is no adverse or additional toxic effects that you can see in that particular animal. So we have seen the bell curve and also the dose response curve. So we will see like how in the toxicological effect, like how it is going to. Okay. So describing here, like there are like different toxicological effects. So toxicity described by exposure duration. So duration has equally important role in the toxicity. It depends on how much period the person or the organism is being exposed to that particular chemical or pesticide. It will determine the toxic effect that a person or an animal will have on it. So toxicity described here, there are like three different kinds of, based on the durations, three main categories are there. One is acute, subchronic, and chronic exposure. Acute in the sense immediate. Immediately you can see it's rapid or immediate effect where it lasts for 24 hours to or less than 24 hours. So it refers to a single dose, but you give it in a higher concentrations. If a pesticide is given in a single dose in higher concentrations, it will rapidly show the ill effects of it. For example, poisoning, chemical spill. If a person for a suicide, in certain cases like suicide, we see that when a person consumes poison, he will there are higher chances of him to die immediately, right? So that is like acute. So immediately you take higher concentrations of the pesticide, then you will see the outcome very early, like the poisoning or death of the person will happen. But in so that is acute exposure. And coming to subchronic exposure, it refers to repeated or continuous. So over the period of time, there will be a development of the symptoms, and that lasts for like a duration of 1 to 3 months. It is majorly seen in some occupational exposures. If a person works in a company or an industry where heavy metals, where he needs to deal with the heavy metals, or a welding person, so there are like certain other toxicities or as well. So in the workplace or due to some occupational exposure, they tend to slowly develop ill effects to that particular pesticide. So that is like 1 to 3 months. So coming to chronic exposure is like it involves a long-term or a repeated exposure to a toxic substance, which is like more than 3 months to years as well. It's a long term, but it will permanently damage the organs or tissues in our body. Uh, and it is like irreversible. You cannot reverse it back. So it's like irreversible, and like certain examples for this are like cancer, liver, kidney failure. So these are all comes under chronic exposure. And depending on this, there is also like toxicity is also characterized whether it is reversible or irreversible. In majority of the cases, it's reversible, and in in acute cases, it can be reversible, but in chronic cases, it is like irreversible, right? So the toxicity of a pesticide can be classified as reversible or reversible depending on the nature and extent of the damage it has caused to an organism's body. So reversible toxicity is an acute exposure, as we've mentioned, to certain pesticides. They may cause temporary symptoms like nausea, dizziness, headaches, or mild skin irritations. So if we, while I've given the example earlier, like if a person takes poison, then immediately if you rush him to the hospital, then there are higher chances for him to recover, right? So that is like reversible. Yes, he has consumed the poison, he has taken the poison, but still, if the treatment is given on time, then there are chances of to remove that poison, and there are chances of that person to live. So that is a reversible one. That is in acute exposure, the reversible chances are higher. Whereas in irreversible toxicity, it refers to a type of damage that cannot be undone. So you cannot do reverse it back. So once the pesticide has caused the damage, the body cannot recover or restore the affected organ or system to their previous state. So majorly it's seen in chronic exposure to certain pesticides, which results in irreversible damage such as neurological disorders, memory loss, motor impairment, cancer, or endocrine disruption. And certain, for example, organophosphates, carbamates, or pyrethroids, they cause reversible toxicity, whereas DDT, arsenic chloridate, they cause irreversible toxicity. So toxicity can be characterized based on the effect they cause. Like the toxicity of pesticide can result in a wide range of effects ranging from mild irritation to severe health condition, including death. The severity of the effects often depends on the type of pesticide and route how they have come into contact with the body, how they have been in the body. So the dose and the duration of the exposure is like they can immediately cause death. That is like in case of high acute toxicity, irritation to skin, eyes, and respiratory system, skin sensitization, like allergic reactions you can see if it came into the contact of the skin. Then mutagenicity can happen. The mutations in the genetic level or the genetic code can happen. Then there are high chance of certain pesticides to be carcinogenic, which causes cancer. And neurotoxicity, the nervous system will be damaged. Endocrine disruption will also happen to the endocrine organs. And reproductive toxicity also leads, which affects the future generations and the reproductive capability of the females and also to the offsprings. So that's the toxicity described by target organ systems will also affect, I mean, it affects different organs of the body depending on the type of the pesticide, the route of its exposure, and how much dose that has been given. So based on all these parameters, different organs will be targeted by the pesticides. Coming to the cardiovascular system, so heart and blood vessels. So when a pesticide is given in higher concentration, then there's chance of altering the heart rate, blood pressure, and if it's above the toxicity, if the toxicity is more, then the damage to the blood vessels can also happen. And in the case of respiratory system, lungs and airways, so irritation of the airways to severe damage to lung tissues can also happen. So it depends on the doses. If it's a lower dose, then just the irritation of the airways will happen. So if it's in a higher doses, then the lung tissue damage can also be seen. Coming to gastrointestinal system and stomach and intestine. So ingestion of these pesticides in food causes gastrointestinal toxicity. If it's in a mild dose, then it can cause mild symptoms like nausea from mild to severe, and life-threatening damage to the gastrointestinal tract. And coming to urinary tract, so kidneys and urinary tract are being affected here. So some pesticides are nephrotoxic, and they can lead to acute kidney injury or chronic kidney diseases. Especially upon prolonged exposure, you get chronic kidney disease. And muscular systems, certain pesticides can cause muscle toxicity, weakness, and cramping. Skeletal system, like bones, endocrine disrupting pesticides can affect bone health over the period of time. Dermal, when it comes in contact with the skin, when the pesticide comes in contact with the skin, then you can see the prolonged skin contact or inadequate protective gear during applications can cause this. Nervous system, so many pesticides are neurotoxic, affecting the central nervous system, peripheral nervous system, leading to both acute and chronic neurological effects. And so we have studied these pesticides, they are very much toxic to the human body, to different target organs. They target different organs and different systems. So here, use of animals in toxicity studies will be seen. So according to the Insecticide Act, it requires pesticide manufacturers formulations to submit. So based on the So according to the Insecticide Act, it requires pesticide manufacturers and formulators to submit data. So we will have toxicity studies, use of animals in the toxicity studies. Insecticide Act requires pesticide manufacturers, formulators to submit the data. And there are also certain boards like Registration Committee of the Central Insecticide Board, which evaluates the data and permits toxicity studies. To evaluate the toxicity of the pesticide, to evaluate the toxicity of a pesticide, the registration committee may permit the manufacturer to conduct a series of toxicity studies. These are studies are critical in determining the safe levels of exposure and understanding the risk assessment here. So advantages of including lab animals as models in pesticide testing are as below. So disease-free animals bred for uniformity are available commercially at reasonable cost. So these are certain advantages of using animals as models in pesticide testing programs. So lab animals can be produced in numbers that is sufficient for toxicological evaluation investigations, and they can be housed in a relatively small space, and they provide accuracy in the interpretation of toxicological data. Use of genetically similar individuals can lead to more consistent results. And the other uses of the using animals as models to study the pesticide testing programs are the relatively the shorter lifespan of the lab animals, which facilitates the observation of effects and diseases associated with lifetime exposures. And diseases like cancer can be predictably observed by using specifically grown or breed animals, which are targeted for particular cancers. And experimental animals reach sexual maturity at an early age, have relatively short pregnancies, and produce large litters, which helps us to evaluate the pesticidal impact on reproduction over multiple generations. And also, the route of entry simulates the human exposure, yielding a relevant understanding of the chemical state and properties of a pesticide inside the body. So experiments can be standardized by monitoring and controlling uncertainties. For instance, food and water quality can be monitored and adjusted as required to achieve standardization, and environmental factors such as temperature, light can be manipulated in case of animals that we use. And commonly used species in pesticide testing programs are, so here when the animals that we use, they are dependent on the study that we make, right? So each study has specific goals. So you have to choose your animals according to the study that you're doing and what are the parameters that are you looking into and what type of toxic compound you're assessing and duration of the study along with the regulatory requirements. So different animals can be used for testing the pesticides, but majorly rodents are used like mouse and rat. They're like very much used and they have high metabolic rate, making them useful for assessing acute toxicity. Mouse, the scientific name is musculus, so they have high metabolic rate and they're like majorly used, like 70% of the animals that have been used for studies will be mice. Then later the rats, that is Norvegicus, they're like well understood biology and short lifespan, reproducibility, and they're like very easy to handle. So majority of the studies will be based on them, on mice and rats. But a few studies like skin and eye irritations, they need larger animals. And for that, it can be seen in albino rats. And also, these are also studied for some birth defects. Guinea pigs used in the hypersensitivity and allergic reaction studies, and dermal irritation and sensitization tests as well. Coming to domestic hens, in the category of birds, they're like sensitive to organophosphorus insecticides, thus used to evaluate nervous system toxicity for this class of pesticides. Dogs are also used, like special beagle dogs are commonly used for long-term subchronic and chronic studies. So this is the table that shows like different exposure toxicities, what are the irritations that they cause, and the species that are used in different studies, like in acute toxicity, the single dose, as we have discussed earlier, the time and all are given in this table. And here we can see like rats, which are used for oral dermal inhalation in case of acute toxicity, mice, rabbits for dermal studies, guinea pigs for dermal studies are used. In case of subchronic toxicity, rats for oral, rats, mice, dogs are used for oral, and rabbits are used for dermal. So these are all the some in vitro and in vivo studies that have been done. So based on the genotoxicity, neurotoxicity, pharmacokinetics, dermal toxicity, and ecotoxicity, there are different species of animals have been used. And coming to the tests used for acute toxicity. So acute toxicity, we know that it is like immediately it will show the effect in higher dosages. So single exposure, high dose or short duration is generally seen here. So to evaluate the harmful effects of a single exposure to a substance, typically at high doses and over a short period of time, then these kind of tests are conducted. So these tests help determine the lethal dose or threshold dose at which adverse effects occur. So basically, there are 6 tests are used for acute testing. One is oral acute toxicity test, which will detect the LD50 of the particular pesticide and the lethality of it. A dermal acute toxicity test, which is like directly applied to the skin in a single dose, typically on occlusion and left for a short period of 24 to 72 hours to see any reaction and assess the lethal dose and any irritation or systemic effects from dermal exposure. An inhalation acute test toxicity test is to determine the lethal concentrations, LC50 of a substance when inhaled over a short period. Eye irritation test acute ocular toxicity. The study observes the degree of irritation, corneal damage, or potential for blindness in case of higher doses. And skin irritation corrosive test is also being done to assess the severity of irritation. Erythema, which causes the redness of the skin. Edema is the swelling of the skin occurs. So it is determined to see that. Acute dermal sensitization test. So evaluation of allergic responses such as skin redness, swelling, or hypersensitivity reactions on skin. So these are all the different tests that are being used to test the acute toxicity and what it causes, like to mild to severe illness or ill effects that it causes. Coming to acute mortality studies. So acute mortality studies are single dose mortality studies. They assess the consequence of short-term high dose pesticide exposure on animal health. And it has pesticides are administered to 3 routes in this, like oral, dermal, and inhalation routes. Oral administration, pesticides are administered orally, where open through a gavus or a tube inserted to the stomach or food or in drinking water to laboratory animals, typically mice and rat. So here you give the pesticides by inserting the tube into the stomach in the food or in the drinking water that they consume and eat. So in that you will orally give the pesticides to laboratory animals, mostly like mice and rats. And in the dermal administration, the pesticide is applied directly to the skin of the lab animals, often at different concentrations. This can include both single dose or repeated or multiple exposure as well, repeated doses. Inhalation administration. So lab animals are exposed to a pesticide in a controlled environment where they inhale the substance in either vapor form or aerosol form. And this is often done using a chamber where the pesticide is vaporized or atomized and animals are exposed for a specific duration. And mostly, this acute mortality studies does not last more than 14 days, but some can extend to evaluate longer term effects based on the studies. Coming to acute irritation and sensitization studies, testing. So pesticides are critical for assessing risks to human health, particularly for occupational safety, where farm workers or pesticide applicators, they tend to get. So the DRACE test for eye irritation and dermal irritation tests are the most established methods that have helped shape safety of the standards, especially in acute irritation and sensitization phase, where eye irritation studies to assess whether a pesticide causes short-term or a long-term damage to the eye. We use DRACE eye irritation test. It's a standard test method for assessing eye irritation and corrosive effects of the pesticides. And it typically involves initial observation, observational intervals, and extended observation. So initially, immediately after you apply the pesticides, immediately you start to observe the reactions and you note down the initial observations or like the reactions of it. Then continuously you will have a specific regular intervals of time. You give it like 24 hours, 48 hours and 72 hours, and you check for any severity of irritation or damage to the eye. And then for an extended observations in case where the pesticide is susceptible of being highly irritating or corrosive. Additionally, the observation periods will be extended to 7 days or longer. That may be conducted to evaluate the reversibility of the damage as well. So this is like a Dre's eye irritation test. When it comes to acute dermal irritation test, so you first initially take a small amount of the pesticide and then it is like applied to the patches of a shaved skin, typical on the back of the rabbit for a set duration of 4 hours. After the exposure, the substance is removed and the skin is observed if there is any signs of irritation or any swelling or anything will be observed here. So observation period, the test involves observing the skin at multiple intervals after the pesticide is removed, generally at 24 hours, 48 hours, and 72 hours. Some studies may extend this observation period if needed to assess the full recovery of or the persistence of the irritation. There are also other studies like dermal sensitization study, like skin sensitization. In dermal sensitization studies designed to evaluate whether a substance or a pesticide can cause allergic reaction upon repeated skin exposure. So this study identifies whether a chemical can sensitize the skin or causing an allergic response upon subsequent exposure. Right, so test method is one of the most common methods for assessing dermal sensitization is a local lymph node assay, which detects immune response after dermal exposure. And another method is GPMT method, the guinea pig maximization test. So induction phase, here we will have different phases like induction phase and challenge phase. Induction phase is for 5 days of repeated exposure. You can see that changes. And challenge phase is 1 day of exposure with evaluation of 24 to 48 hours or later. And the observation is the allergic response is typically evaluated within 24 to 48 hours after the challenge phase. That is about the dermal sensitization study. Coming to acute non-delayed neurotoxicity testing. It is acute, like it's immediate, non-delayed neurotoxicity. Without any delay, it will cause the nervous system effect, immediate toxic effects of a pesticide on the nervous system. So after a single exposure itself. And it assesses both the central and peripheral nervous systems for any signs of neurotoxicity within a short term after exposure. And test method is the acute neurotoxicity study is typically used for assessing acute non-delayed neurotoxicity. It is conducted by administering a single dose of a pesticide via dermal or oral or inhalation through any route. And acute delayed neurotoxicity testing. So we have seen non-delayed means immediately toxic effects will be seen on the nervous system, whereas in a delayed acute delayed neurotoxic testing focuses on detecting neurotoxic effects that occur after a lag time. I mean, it will give some time. Then after that, you can see the effects. So following initial exposure with no immediate signs of toxicity. But however, the toxicity is particularly important for substances that may affect the nervous system with a delayed onset. Coming to acute non-delayed neurotoxicity testing, here we do evaluate the immediate toxic effects of a pesticide on the nervous system after a single exposure. It assesses both the central and peripheral nervous systems. And the tests that have been majorly used are the FOB test, which is a functional observational battery. It's a standardized set of tests used in neurotoxicity studies to assess general neurological functions, including the motor activity, sensory perception, and reflexes. So what is motor activity? The functional observational battery includes tests that assess both spontaneous motor activity, that is general physical activity, including ambulation and rearing, and more specific motor coordination tasks. Example, balance, gait, rating reflex, and all. That is commonly used both in acute neurotoxicity and delayed neurotoxicity studies to evaluate the effects of the pesticides. And the time and duration of it is like acute neurotoxicity. FOB and motor activity are assessed at various time points immediately after exposure, typically at 1, 4, and 24 hours after a single dose. And delayed neurotoxicity, the same tests are administered during the initial observation period and again at 7 days, again at 21 days, like 2 weeks and 3 weeks after exposure to evaluate whether delayed neurotoxic effects have been developed or not. So we have done with the acute toxicity studies. Then we'll be dealing with the subchronic toxicity studies. So it determines the safety of a pesticide during repeated moderate duration exposure. So you don't give high doses and you see it for a shorter time. But in subchronic toxicity studies, what happens is that So you give like moderate exposure, moderately you give the drug, you don't give high doses, and then you check how it is happening. So this test provide very important information about the intermediate dose and moderate duration exposure to pesticides, helping to identify the NOEL and LOEL and specific target organs per toxicity. And standard durations of the studies are like 90 days for 3 months, and the intermediate dose is typically selected to detect sublethal toxicity, providing insight into long-term health risks of pesticide exposure. In this study, there are like different modes like oral toxicity, dermal toxicity, inhalation toxicity, and neurotoxicity. Oral through food or water, you will give the pesticide to animals like rats, mice, and you will see the duration for 90 days, and then you'll find any, you'll check for any body weight or clinical chemistry or organ pathology, like any damage to the organs or histopathology, you check in that. And coming to dermal exposure, so dermal, the skin will be exposed to the chemical, and the animal models that are mostly used are like rats, guinea pigs, and dogs. It will be also for 3 months. Uh, I mean, from it will be like below 3 months, the subchronic toxicity studies, and there you get to observe the skin irritation, systemic absorption, and organ pathology. Inhalation, again, through aerosols, it can be exposed. The exposed route is through aerosols, and rats, visitors, rats and two in the rats, special species like visitors, progeroли are used. Mice, dogs can also be used for inhalation subchronic toxicity studies. So there you'll get to see the respiratory function, histopathology, and organ pathology. And coming to the neurotoxicity, oral dermal inhalation will be seen in rats, mice, and dogs, and that is also for up to 90 days, and where you get to see the motor activity, reflexes, cognitive function, histopathology of the nervous system. Here. And coming to the chronic toxicity testing. So we are done with the acute toxicity, subchronic toxicity, and now we will be dealing with the chronic toxicity testing. It's essential for evaluating the long-term effects of pesticides when animals are exposed to low doses over extended periods. So to assess the long-term health effects of the pesticides, we use chronic toxicity studies or testing, and it is used to identify target organs affected by prolonged pesticide exposure, determining the no observed adverse effect level and lowest observed adverse effect level. So we can detect that easily in chronic toxicity studies to ensure that the pesticide can be used safely without causing chronic health problems in humans, animals, or the environment. So there is a term like maximum tolerated dose. It is the highest dose of a pesticide that can be administered to an organism which will not cause any damage, that is like an without causing any irreversible damage or significant adverse effects in the animals. So a dose, that is the maximum pesticide dose that you can give to an organism which will not cause any lethal effect or which will not damage the organ completely, is called maximum tolerated dose. And here the chronic toxicity test again will be like chronic oral toxicity, dermal toxicity, inhalation toxicity, and neurotoxicity. Again, here you will give in oral toxicity, it is like similar in oral. However, whether it is like acute, subchronic, or chronic, you will give them in the food or in the drinking water. But what will vary in this acute, subchronic, and chronic is the duration, right? Duration, if it's a chronic, then it will like 1 year or more than 1 year also will take place in few studies, and those levels will also vary. So in the chronic toxicity, we will give low to intermediate, then slowly the higher, the maximum it can take will be dealt with in the chronic toxicity. So in oral toxicity, the endpoints to meet is like we check the body weight, organs weight, histopathology, clinical chemistry, cancer, reproductive, and neurotoxic effects in case of oral toxicity. In chronic dermal toxicity, then we check for skin irritation, organ damage, systemic toxicity, and histopathology. And chronic inhalation toxicity, we check the respiratory effects, organ damage, systemic toxicity, and cancer. And chronic neurotoxicity, we check the motor activity, cognition, reflexes, neurotoxicity, histopathology of the nervous system. Not only this, we know that pesticides affect different organs and different systems in our body. So there is also like reproductive toxicity testing. So we do toxicity testing for reproductivity as well, which is majorly involves two categories like developmental and reproductive. And in developmental, teratological tests will also include, and in reproductive, we will add the fetal malfunctions as well. So in the reproductive toxicity testing of the pesticide, it's a critical area of safety evaluation to assess the potential effects of the pesticide exposure, which is very much involved in fertility, reproduction, and development. So as we have talked, two types of tests focus on determining if exposure to a pesticide can cause developmental or birth defects or reproductive toxicity, potentially affecting the future generations. So the type of the study is developmental toxicity, where you get the focus area here is teratogenesis, that is like a birth defects or abnormal fetal anomalies can be detected here. The fetal development will be checked here, and it will be done for a duration of 14 to 21 days of gestation. And majorly the main species that have been involved in the developmental toxicity are rats, mice, and rabbits. Here you get to see the external and internal malfunctions, growth retardations, and also the birth defects can be detected. And coming to the reproductive toxicity, it is depending on the fertility, mating, and reproductive function. So here majorly the female rats, mice, and dogs will be considered for this reproductive and developmental studies. And in reproductive toxicity, the duration will be like 60 to 90 days in case of acute exposure. So mating will be done. After the successful mating, then the pregnancy or the fertility or the offspring survival and will be studied in the reproductive toxicity. Right, so they are like a generational studies. So you get to see in the reproductive toxicity, what you get to see is the generational studies. You see, when you take the healthy female rat and also the male rat, then once the mating is done, then the successful pregnancy will be there. So after that, you give the same food with the with a special compound that you want to check, and then you see that pass on to their generations. Then this generation again they go for successful mating, again they make offsprings, and in the second generation as well you tend to see. So the main advantage of using rats, mice, or these rodents is that they are like early they mature and they help us, they give like more litter, so they help us to study the generational studies and how how these pesticides will have effect on the future generations or upon the generations, like how they act. Okay, so the multi-generational reproductive toxicity will fall into this, like the fertility, reproductive functions, offspring development, and organ histopathology. And it is an extended study, definitely. If you have to study the generational studies, then it can last for 1 to 2 years or more. Coming to teratogenicity studies, the fetal malfunctions or growth restrictions can be studied, and it also falls within the gestational duration, like 14 to 21 days, and specifically seen in rats and rabbits, where you get to see malformations, developmental delays, or any birth defects or fetal anomalies can be seen. The duration of these studies varies depending on the scope and depth, with developmental toxicity typically assessed over 14 to 21 days. So during pregnancy, reproductive toxicity spanning 60 to 90 days, and multi-generational studies lasting up to 2 years will be studied to assess the long-term effects across the multiple generations. So after reproductive toxicity studies and chronic toxicity studies, we'll get to see the genetic toxicology here, which involves the study of, in the genetic toxicology, you'll see how a pesticide can actually affect genetic material, be it an RNA or DNA, how it can affect the genetic material, leading to changes in the chromosomes and the structural changes in the chromosomes, the chromosomal aberrations, and germ cells and somatic cells. If the chromosomal aberrations, you get to see the change in the structures that can occur due to environmental exposure, including pesticides and chemicals. So which will also be numerical and structural. In numerical, you tend to see that the chromosomal number will be abnormal, and euploidy, where cells have an abnormal number of chromosomes. And coming to the structure of the chromosomes, so there will be certain deletions, translocations, inversions, duplications, and additions. So anything can happen that ultimately lead to the change in the structure of the chromosome in the genetic level. And with the germ cells, the gametes, the germ cells like sperm and eggs are crucial for passing genetic information to the next generation. So pesticides can induce genetic damage in these cells as well, so potentially leading to mutagenesis, that is the mutation in the genetic level where it gets inherited to the next generation and next offsprings as well. Somatic cells, so these are not the reproductive ones. So the somatic cells, they help us to build the bodies and tissues and organs, and they are not involved in the reproduction, and they majorly cause cancer or developmental abnormalities. And some of the examples of the pesticides that cause chromosomal aberrations, DNA strand breaks, and mutagenic effects are organophosphates and carbamates and pyrethroids. They affect the cell division, leading to chromosomal aberrations. Organochlorines are long-lasting effects of the genome, causing mutations and chromosomal damage. So this is the genetic toxicology. So now you know that these pesticides, they cause changes in chromosomal level. They also cause changes in the germ cells. They also cause changes in the somatic cells. So how do you test them and how do you see them? Is that we have in vitro and in vivo assays to that. What are in vitro and what are in vivo assays? In vitro assays are assays that you do work in the lab in the test tubes using cell lines of the bacterial, fungal, or human cell lines that you use. Whatever you do in the tube, they are like basically like in vitro studies. In vivo assays are like when you use a whole animal to study the toxicity of the pesticide or whatever it is. If at all you use a whole animal, then it is an in vivo study. Okay, so we have both in vitro and in vivo assays to evaluate the mutagenic potential, with each method targeting specific genetic endpoints such as gene mutations and chromosomal aberrations. Chromosomal aberrations here. So bacterial gene mutation tests like Ames test can be studied. In the Ames test is one of the most widely used in vitro assays that have been used, and this was named after the honor of the scientist Dr. Bruce Ames in 1973, who developed this test using the bacterium Salmonella typhimurium. It is proven efficient and reliable method for testing chemicals for their being mutagenic and carcinogenic. So in Ames test, bacteria are treated with a chemical with and without metabolic activation and grown on agar plates in such a way that only the mutated bacteria will grow. So bacteria colonies are counted after 2 days and compared to the number of colonies growing in controls. So if you compare them, then you'll tend to know that whether it is grown or not. So positive mutagenesis response occurs when the number of colonies with a pesticide treatment is 2 to 3 times higher than the number of colonies from the untreated bacterial cultures. So the test involves exposing bacteria to a pesticide or metabolites, assessing the frequency of the reverted mutations, that is the mutation that reverses a previous mutation in the bacterial genome. So the bacteria using the M tests are typically auxotroph, means they cannot grow without that particular specific nutrient. So if at all the mutation occurs, their genome can restore their ability to grow even without the presence of the particular nutrient. So this indicates that there is some gene mutation. In vitro mammalian gene mutation tests like CHO, GPRT analysis can be done. So here CHO means the Chinese hamster ovary cell line. So this is a cell line. Cell line studies we do, cell culture studies we do to test particular compounds. So these are in vitro studies. Basically they are done because you don't have to initially to confirm whether this compound is toxic or not. You directly don't go and do it on the animals, as it's like very tedious and like it is not even ethical to sacrifice and unnecessarily sacrifice an animal. So initially you have to do certain assays and certain confirmations and certain studies in vitro without harming any animals according to the ethical committee. So majority of the tests will be done initially in vitro, and then upon confirmation and for further studies and for more relevant and physiological and biochemical studies, you can definitely go for smaller animals. In vitro mammalian gene mutation test, here Chinese hamster ovary cell lines is used with the hypoxanthine gluconine phosphoribosyl transferase gene. It's a mammalian cell-based test used to assess gene mutation in mammalian cells. In this test, what happens is that Chinese hamster ovary cells are exposed to a pesticide, and if at all any mutation in the HGPRT genes is detected, the HGPRT gene is responsible for converting certain purine analogs into a functional nucleotide. So it is used in a Salvage pathway because it converts certain purines to functional nucleotides. So a mutation in this gene prevents this conversion, which can be detected by the inability of a cells to grow in the presence of a specific purine analog. Right? So basically what does this gene HGPRT do is like it converts certain purines for its functional nucleotides. If that is not there, if the gene is defective, then it will not be able to convert it. So there you cannot see any purines. So in that way, you can say whether the gene mutation has occurred or not. In vitro mammalian chromosomal aberration test, it is also done in CHO cell lines or lymphocytes. Where the pesticides will be exposed to is this. So the cells are cultured and then the pesticide is exposed to the cells and then examined for chromosomal changes. So the types of chromosomal damage are evaluated include breaks, so you can see the changes in the chromosomes if there is any break or translocation of the genes or any deletion of the gene occurs or any micronuclear formation is seen, then you can know that the cells are typically harvested during metaphase. Metaphase stays in a cell division which chromosomes are most visible. In the metaphase, you can clearly see the chromosomes under the microscope. So at that stage of the cell division, you will harvest the cells and then you will observe for any breaks, translocations, or deletions or micronucleate formations. If at all you come across such deletions or such changes, then you'll confirm the chromosomal aberration has happened. In vivo mammalian chromosomal aberration. So we have seen in vitro using Chinese hamster ovarian cell lines or lymphocytes. Then in mouse as well. In vivo means in the whole animal. In the mouse also, micronucleus test is available to check the chromosomal aberrations in it. So how it works is mice are treated with pesticide and their bone marrow is collected or harvested at specific time points and the presence of the micronuclei will be counted. So micronuclei form when chromosomes are parts of the chromosomes are not properly incorporated into the so if this proper cell division does not occur due to any of the translocations or deletions, then definitely there will be a micronuclei formation in the bone marrow. So by that you can say that yes, there is some chromosomal structural deformation that has occurred. So here it is just a gist of the essays that we have seen earlier. So this is the table that indicates the gist of the essays. And coming to the methods of testing chemicals on insects, so there are like different methods of testing. There are like topical methods, injection method, dipping method, contact residual method, feeding and drinking method. So in the topical method, so in a topical, definitely you know that when a chemical comes in contact with the skin, the toxicity will occur. The advantages of this is that precise doses is used and good for contact insecticides. If the insect comes into the contact, then this you can see the insect is then observed for any adverse effects such as mortality or behavioral changes. In the topical application method, in the injection method, what will happen? A chemical is injected directly into the insect's body, often into the hemocoel, the insect's blood cavity. Thus bypass of the insect's external defense mechanism and digestive system, providing a direct route for the chemical to enter the insect's internal structure. In the dipping method, you'll submerge the insect into the solution containing the chemical of interest. This is typically used for testing chemicals that are intended to be observed through the cuticle or when assessing the effects of the treatment like insecticides on egg cases or larvae. A contacted residual method, so this is by through spray or dust or coating on the surface, they come into contact. Feeding and drinking method is like through food or water, they come into contact. And in feeding method, you have two types, like unlimited feeding and limited dose feeding, where you give constant access of pesticide-treated food to the insect for a specified time, allowing them to feed freely and consume varying amounts of the chemical, where you will not have a specific amount that has to be given for an insect to consume. But in a limited dose feeding, insects are allowed to feed on a treated food source for a limited period of time during which they consume a specific amount of the chemical. In a drinking method, similar to feeding, this involves providing the insects with a pesticide-treated solution that they drink over a specified period of time. So here, over a specified period of time, you tend to see this. So this is the gist of it. I guess you can see. So this is the gist of it. The types of exposures, the advantages, the disadvantages that are involved in the method of testing of the chemicals of this particular insects. So we are done with the methods, tests, and all of this. So now we have the statistical part of this. Like after doing all this, about the statistically how you have to represent the data, then definitely we will go to the probability unit analysis. So probability analysis is a specialized form of regression analysis which is applied to binomial response variables. So binomial in the sense it is based on one or more predictable variables like whether it's death or life. So here you will see whether it's like a death or life or the percentage when susceptibility of any population to a poison is normally distributed. It is usually assessed by constructing a dose mortality curve by plotting the dosage against the percentage mortality. So here you can see that two possible outcomes will be there for a probability. So the probability unit is here termed as a probit. Probit is nothing but the probability unit of analysis. The key difference from logistic regression is that the probit model assumes that error terms follow a normal distribution, whereas logistic regression assumes a logistic distribution. So here, probit analysis is a parametric procedure and that relies on the linear regression, and ideally what happens is that in a linear regression, the relation between the dependent variable and the independent variable is assumed to be linear, meaning that it can be represented by a straight line. However, the data here on probit analysis involves the linear regression model. It works on the linear regression. Linear regression is nothing but a statistical method that is used to model the relation between a dependent variable or the outcome or a response variable on the independent variable where the predictors or the features will be seen in the independent variable. In a linear regression, um, in a linear regression, the relation between the dependent variable and the independent variable is assumed to be linear, making that it can be represented by a straight line. So the equation of the linear regression will be y equal to a plus bx plus e says that the dependent variable y is a linear function of the independent variable x. So in a simple linear regression, you can see that it models a relation between two variables. One is a dependent variable, that is y. That is y. It is a dependent variable. And another one is the independent variable x. So y is the dependent variable that is the outcome you want to predict. X is the predictor which predicts this outcome. A is the intercept value. A is the intercept value and B is the slope. E is the error term. Error term. The difference between the observed and the predicted values of the Y is the error term. Okay. So linear regression helps us to understand how the independent variable and influences the dependent variable. For example, if in an easy terms for us to say, for a better understanding, we can say like this: like if you're trying to predict a person's salary. Okay. So here salary will be a dependent variable. What is it dependent on? It is dependent on based on your experience, right? So your experience is an independent variable. So you get your salary based on your experience. So here salary is a dependent variable. The coefficient slope B will tell you how much salary. So here B will tell you how much salary increase for each additional year of experience you get. So I'm clear. So in general, data derived from toxicity tests are generally proportion or percent response data here. So because there are differences in susceptibility among individuals with a given population, the dose response curve has a sigmoidal shape. So initially the dose response curve, we tend to see it in a sigmoidal shape here. So here it is based on the dose response curve, right? In the dose response curve earlier, what we have seen, like if you keep on increasing the dose, then the response or the how the population is responding will also depend on this. So as it is in the S shape, we call it as an sigmoidal shape or the sigmoidal. Okay. And when the mortality frequency of each group is plotted, the bell-shaped curve happens. So we have earlier studied about the bell-shaped curve or the normal distribution, where I told you like maximum persons will be, the majority of the population will fall into this empirical rule, where the more least, the majority of the sensitive population will fall under this, and the highly resistant will fall under the tail of this, where the majority of them will be this. So here you can see the mortality frequency of each group. If you want to plot, then you have to go for the normal distribution of this. But when you have to convert this into this, so if at all you want to convert, the probit approach is generally used, in which the percentage response curve is converted into units of deviation from the mean. So in this, despite the distinction between dose and response concentrations, so here depending on the units of the probit values, so here you wanted to plot a straight line. So if you consider the probit values, then you get a straight line here. Because there is a difference in susceptibility among individuals within a given population, the dose response curve has a sigmoidal one. So the majority of the cases or the things, in other words, some of the animals are susceptible and will succumb to the chemical at a fairly low doses level. The majority, about 68%, will succumb in a dose response that falls here in the -1 standard deviations. So here the probit 5 is defined as the 50% response. So if you consider the here 50% means it is probit 5 and a probit 6 value. So we can better understand this. If at all you convert the standard deviations from the mean into a probit scale, so the ideal is to convert proportions to probability units on the LT scale. So here you can see that the probit of 5 corresponds to the midpoint of C50 or C6.64 correspondence. So this is the Finney, Dr. Finney had given this table, probit table. Here, this is the percentages, and these are the probit units. So if you get like, if it is a 5% correspondence, then you call it like this. Let us see here clearly. So where is the 6.64? So here it is like 6.64. The probit unit is 6.64, right? So if you get the concentration 95, if the concentration here, if the concentration is like 95, so check here, 90 to 100 you have, no? So here, if here the concentration is 95, then here you have to check, like you have to go into this, the percentages in tens, and then in 1 unit you have to place this, and here you have 90. From 90 you have to go to 95, come down to 90. So here you have, if the C is 95, then definitely the probit value is 6.64. And if it is C5, then it is 3.36. C5 is here. So if it is just the percentage of the concentration is like only 5, then it is 3.36. So based on this, what you get is the concentrations of the proportions will be converted into probability units. So in this, the conversion will happen into the probability units. The probit 5 corresponds to the midpoint of a concentration, and 6.64 corresponds to 695. The conversions of proportions or hit rates to probits can be carried out using the table provided by Finney, where P is the proportion of the hit rate. Hit rate is nothing but the probability that has been calculated from the raw data. And you can get to apply this probit analysis here. So probit analysis is commonly applied in toxicology, dose response studies, to analyze the relation between the dose of a substance such as toxin, drug or chemical and the observed biological response. Example, mortality, morbidity or other physiological effects. And also through probit analysis, you can get to know that you can estimate the lethal dose 50 and ED 50, effective dose 50 as well. That we have read about them in our early classes. Like lethal dose is the 50% of the population will get killed. That is the lethal dose, and effective dose is like it's also the important factor for detecting the how much of the dose is like effective for 50% of the population. So that can be done by probit analysis. So probit analysis can be done on graph, or it can also be done using an Excel formulas Or you can also do the probit analysis using SPSS softwares as well, some statistical softwares, which you if at all you want to analyze the dose response curve, it helps to model non-linear dose response relationships where the effect of the substance increases with the dose. Like we have seen the dose response curve, if you give more concentration, then the severity will also be more. That is a sigmoid curve you have seen earlier. And also to affect the effectiveness of the pharmaceutical pesticides, like in the probit models, it helps to determine the it helps to determine the ideal doses that maximize the therapeutic efficacy while minimizing the toxicity. And also it is used to estimate the risk and hazard assessment. So probit analysis is also used to in risk assessment and regulatory toxicology to predict the probability of an adverse effect such as cancer, organ damage, etc. at different exposure levels. It helps the regulatory agencies to frame regulatory guidelines in such a way that the exposure limits by modeling the relationship between dose and probability of the adverse outcome is relied upon. So which can be used for setting safety standards before using the pesticides. And also comparison of different toxic substances can be done with the known substance that is being available or with the known values of the probit that has been done for different pesticides, where this is especially useful for evaluating new chemicals, pharmaceutical agents, or environmental contaminants in relation to known substances. And evaluating the chronic versus acute toxicity, yes, you get to evaluate whether it is like immediately toxic to the person or the animal, or whether it is like it is like over the period of time it causes any severity or damage that can be evaluated by using this statistical method. And it's also used in analyzing sublethal or subclinical effects. In addition to lethal outcomes, probit analysis can also be used to study sublethal effects or other types of biological responses to exposure, such as growth inhibition, changes in the reproductive success, or behavioral alterations. So this type of analysis is particularly useful in environmental toxicology, where sublethal effects can have long-term ecological impacts. And some examples to process the probit analysis in toxicology is data collection, that is gather data from toxicity testing, where you record the dose administered and the response observed. So you have to collect the data. So whatever the dose you are giving or whatever the dose that is being administered to that particular animal, equally you have to see the response of it as well. So basically probit is a binomial or a binary outcome, right? So you have to see whether the animal is dead or alive. If so, so what is the lethal dose of it? So at what concentration the LD50 is being caused, or at what saturation, at what plateau stays, or at what concentration there is a plateau stays, and where the animal is dead or whether it is alive after attaining that or not. So that all has to be collected. There has to be a recording of this. And fit the probit model is like use probit regression to fit a curve to the data, which will allow you to predict the probability of a specific response at any given doses. So you will get to know if you give 5 mg, for an example, if you give pesticide 5 mg, that is like a lesser dose, then it is like no adverse effect, you can see that. But immediately at 10 mg, there will be a lethal effect or not, or the low adverse effect, or little symptoms can be seen. Whereas in at 25 mg per kg of the body weight, there can be a LD50 or a lethal dose 50, which kills 50% of the population. So in order to know how much concentration is causing no adverse effect, or how much concentration is causing low adverse effect, and how much concentration is causing LD50 lethal dose, and how much is the maximum doses that has to be given, and at what concentrations the particular drug is reaching the plateau, that can be seen by using the probit model, right? And you have also have to interpret the results, like determine the key toxicity parameters such as LD50, slope, and threshold. Threshold is like up to certain amount of the pesticides, it will not cause any effect, but upon crossing the threshold, that will show signs or ill effects. So the threshold can be known, and use these results for safety assessment, regulatory purpose, or further experimental designs. So with this, we have concluded the second chapter, and we'll just uh, yes. We'll just see the questions that are like mostly important, the model examination questions that we get from this. So as we have done with this chapter, so we'll just see the model questions here. So the questions can be like, what are the advantages of employing lab animals as models in pesticide testing programs? Like we have given like 10 points or so you get questions like the advantages of employing lab animals as models in the pesticide testing programs, and also you can also get questions like explain Ames test or explain gene toxicology test or mutagenicity test. So certain things can also be asked based on the test. And why are the percentage mortality converted into probate values? So why you have to convert the percentage mortalities to probate values and the doses into log values for computing the toxicity values? And what are the various acute tests for assessing toxicity of poisons? Like acute tests, we have like oral, inhalation, and dermal. We have seen like neurotoxicity. So that can be an essay question that you can get for the what are the various acute tests for assessing the toxicity of the poisons. And what do you understand by probability analysis? Probability is a probability units can also be questioned here. And coming to the reference books, Finney Probit analysis from Cambridge, England, Cambridge University Press is can be referred for this. And Hodgson, a textbook of modern toxicology by Ville the Science, the third edition, can also be referred. It can also be a reference book here. And come to toxicology of insecticides by Matsumura is also recommended book for your reference. And the toxicology and biochemistry of insecticides by you is also a good one to refer here. So in the second chapter, we have dealt about the introduction of the evaluation of insecticides toxicities, evaluation of the toxicity, toxicological evaluation in higher animals, the concept of the bell-shaped curve, the relationship between the dose response curve and the concept of the dose response using the dose response curve, the adverse toxicological effects, the toxicity described by the exposure durations, toxicity characterized as reversible or irreversible, and toxicity characterized by the and the uses of animals in toxicity studies and the tox testing of the evaluation of the pesticides and methods of testing chemicals on insects and robot analysis we have done. And tomorrow we'll be dealing with the third chapter, the insect